Hello everyone, welcome back to this RSET training on monitoring water quality in lakes and coastal regions using stream. Today it's part 2 on introduction to a machine learning model to estimate water quality parameters based on satellite observations. We have a guest speaker today, Dr. Ryan O'Shea, and he will be talking about the machine learning model. Last week, in part 1, we saw background, overview and demonstration of Stream web tool and API, where Stream is satellite-based tool for rapid evaluation of aquatic environment. We had a guest speaker, Mr. William Wainwright. He showed us that Stream uses Landsat 8 and 9 and Sentinel-2 A, B, and C to obtain water quality parameters, which include chlorophyll A concentration, total suspended solids, and Secchi disk depth. Area covered are coastal estuaries and inland lakes in the US. The data are available at 20 to 30 meter spatial resolution. And we saw that Stream API allows search and download of multiple images for an area of interest. We saw example of selection of water quality parameters using Stream, map, and make time series of water quality parameters using QGIS. Example shown here, we saw Pyramid Lake, and this map shows chlorophyll A concentration, high resolution data that shows pattern and how chlorophyll A is distributed within the lake in different parts that can be seen here. We downloaded multiple images of chlorophyll A concentration as well as TSS in Chesapeake Bay and made time series using QGIS. Today now we will focus on introduction to a machine learning model to estimate water quality parameters based on satellite observations. This is the model that's used to derive water quality parameters in Stream. Just a note here, there is a homework posted today on our website and it will be due on 10th of March. And the certificate of completion will be awarded to those who attend all live sessions and complete the homework assignment before the given due date. So we'll start with today's session. Our objectives today are that by the end of part 2, you will be able to become familiar with a mixture density network or MDN based model used for deriving water quality parameters from satellite observations in Stream. Recognize how to select and process satellite images for deriving water quality parameters in a water body of interest, and recognize how to access the MDN model code for deriving water quality parameters in the water body of interest. Here's the outline for today. We will start with a very brief review of pre-processing satellite images for inputs to the MDN-based model. Then we will have an overview of the MDN-based model which is used in Stream. And we'll have a demonstration of access and application of the MDN model code for deriving water quality parameters from satellite data. Our guest speaker, Dr. O'Shea, will use examples to demonstrate how to use MDN code. He will use Sentinel-3 OLCI images and PACE OCI images to show water quality parameters derived from this model in San Francisco Bay and Lake Erie. We'll have a brief review of pre-processing of satellite images. Now, satellite sensors measure top of atmosphere or TOA radiances, and these radiances result from a combination of surface and atmospheric conditions, including the effects of clouds and aerosol particles. As seen in this figure, TOA radiances as a function of wavelength are shown here, and contribution is pretty large from atmosphere and also from surface, including this green curve that is contribution from water surface. So this water leaving reflectance that depends on backscattering and absorption of radiation due to materials in water that is optically active, as shown here, that the water leaving reflectances, they depend on what is going on in the water, and these are the reflectances they are used to estimate water quality parameters. To get water leaving remote sensing reflectances or RRS at different wavelengths, we have to start with satellite observations of TOA radiances and apply atmospheric correction schemes to get RRSs. Now here there are a number of schemes or atmospheric correction algorithms listed. NASA has a couple: NASA Ocean Biology Processing Group algorithm and NASA Aquaverse algorithm, which is used for stream. There is Ecolite and Polymer. These are some of the schemes generally used by the community to do atmospheric correction and get remote sensing reflectances. So we would start with satellite images, which is level 1 top of atmosphere reflectance, and then find level 2 remote sensing reflectances that then they are used to estimate water quality parameters. So to summarize, to get remote sensing reflectances, we'll select a satellite and sensor and start with level 1 satellite data. Then use appropriate atmospheric correction algorithm to derive level 2 water leaving reflectances, and then those can be input to the MDN model that we will see in a few minutes. Here we're talking about NASA Sea Earth Atmosphere Data Analysis System, or CDAS, has Ocean Color Science Software, or OCSSW, that can help, that has algorithm for atmospheric correction and can be used to get level 2 remote sensing reflectances. There are a couple of RSAT trainings. They demonstrate how to use CDAS OCSSW to do atmospheric correction and get remote sensing reflectances. So you can refer to these for more information. And CDAS is an open source software from NASA and can be downloaded, and you can find information from NASA Earth data and search with CDAS and you can get more information, or you can refer to these trainings for more details. We saw this table last week, all the satellites and sensors which are used for water quality monitoring. And here are the sources where you can start with level 1 data. You can obtain level 1 satellite images from these satellites, and so corresponding links are provided here. These are the data portals where you can download images from these satellites and sensors. With that, we'll start with today's session on overview and demonstration of MDN model for stream. Just a note here, please put your questions in the questions box and we will address them at the end of the webinar. And feel free to enter your questions as we go. We will try to get to all the questions during the Q&A session after the webinar. The remainder of the questions will be answered in the Q&A document, which will be posted on the training website about a week after the training. With that, I'll introduce our speaker for today, Dr. Ryan O'Shea. Dr. Ryan O'Shea is a senior research scientist at NASA's Goddard Space Flight Center. Ryan received his PhD in Mechanical and Oceanographic Engineering from the Massachusetts Institute of Technology and Woods Hole Oceanographic Institution joint program in 2021 while he was under a National Defense Science and Engineering Graduate Fellowship. Dr. O'Shea's thesis, titled Computational Approaches for Submeter Ocean Color Remote Sensing, focused on the theoretical limitations, practical considerations, and potential future avenues for deploying lightweight hyperspectral cameras for ocean color remote sensing from low-altitude platforms. Currently, Ryan is working on refining freshwater and ocean water products at NASA Goddard. So we invite Dr. O'Shea to present about the MDN model used in stream. Ryan?
Hi everyone, my name is Ryan O'Shea. I'm a member of the Freshwater Sensing Program at Science Systems and Applications, Inc. This program consists of myself, Arun Saranathan, Akash Ashapur, Will Wainwright, and Brandon Smith. It was formerly headed by Nima Palavan, who's moved on to NASA HQ. Today I'll be presenting on monitoring water quality in lakes and coastal regions using stream. Part 2 focuses on introduction to a machine learning model to estimate water quality parameters based on satellite observations. On this slide, I'm going to briefly take you through freshwater and littoral imaging from space, showing the mixture density network supported sensors. So sensors in red are just supported by the MDN, in blue they're supported by Stream, and in green they're coming soon to Stream. In 2000, or actually the end of 1999, MODIS was launched. MODIS is a multispectral instrument which can capture at kilometer scale resolution with a near 1 to 2 day temporal resolution. This satellite sensor has lasted for the last 26 years, so it can produce time series and climatology of given regions. This is supported by the MDN. VIRS was also launched in, I believe, 2012. It achieves about 750 meter spatial resolution and again, 1 to 2 day spatial revisit. It has very similar capabilities as MODIS, and we have models that support both of these and show generally pretty harmonized retrievals from both. We also support Landsat 8 and 9. These are the models that you saw as part of part 1 that are included on Stream. These sensors have fewer spectral bands, but much finer spatial resolution, up to about 20 meter spatial scale, so that we can start to look at fine scale inland water bodies using these sensors. Also supported on Stream are Sentinel-2A and 2B with similar spectral resolution as Landsat and similar spatial resolution. However, the cost for both of these sensors is temporal resolution, with Landsat having a temporal resolution of about 8 days, whereas Sentinel has a resolution of about 5 days. However, when you start to combine them together, you can improve the temporal resolution. So we already have Landsat 8 and Sentinel-2 supported on Stream, but coming soon to Stream, we'll also be adding in Sentinel-3A and Sentinel-3B, for which we already have mixture density network models available. These have temporal resolutions of about 1 to 2 days, higher spectral resolutions, which allow for improved product accuracy in these optically complex inland and coastal waters that we're sensing in, and spatial resolutions of about 300 meters. So by combining the Landsat 8, Sentinel-2, and Sentinel-3 imagery, we can get higher temporal, spatial, and spectral combinations. As additional sensors are launched, we'll also be adding these into Stream. We also have mixture density network models available for commercial satellites like Planet's SuperDove, which achieves meter scale spatial resolution. We also support hyperspectral instruments, so we develop models for HICO, the hyperspectral imager for the coastal ocean, as well as PRISMA and EMIT. We're currently funded by EMIT as well as the PACE Science and Applications team to further improve our products for hyperspectral imagery. The PACE model that we have available supports a wide range of products leveraging the full hyperspectral data. So not only does it support the standard chlorophyll, TSS, and CDOM products, but also inherent optical properties like absorption due to phytoplankton. And PACE allows for 1 to 2 day temporal resolution and about a kilometer spatial resolution. We were also funded as part of the Landsat science team to make models in advance of Landsat Next, which should be launched around 2031. So this large set of sensors that we have developed models for are radiometrically capable for sensing in aquatic regions. The combined observations can get us towards daily observations if we're able to leverage estimates from all of these sensors combined, but they have different spatial and spectral capabilities, which allow us to sense in a wide range of spatial resolution regions, so we can sense in inland as well as coastal waters. The trick, however, is combining these estimates from these different sensors to get a better understanding of global inland and coastal waters. To do that, we need a robust workflow for producing seamless products over fresh and coastal estuaries. To do that, we plan to use a machine learning model, and to train our machine learning model, we need a representative data set. So we've assembled a large data set of a variety of different in situ products, including chlorophyll A, total suspended sediment, as well as color dissolved organic matter. This dataset builds off of Moritz Lehmann's Gloria database, which is cited in the bottom right. It has, at this point, we have about greater than 10,000 globally distributed in situ measurements. These include those biogeochemical parameters I was talking about before, as well as inherent optical properties from inland and coastal waters. As you can see from the map, about 20%, about 80% of this data set comes from North America as well as Europe, and 20% from the rest of the world. Shown here, we have histograms for chlorophyll A, PC, TSS, and CDOM from that globally distributed in situ data set. And these histograms show the wide range of different optical conditions we have represented in the data. So if we just look at the chlorophyll A histogram in the top left, we have about 6,000 measurements, and they span about 4 orders of magnitude. So it represents anywhere from oligotrophic to eutrophic waters quite well. We also have about 1,000 measurements of PC in the top right. PC stands for phycocyanin, which is another pigment, but this pigment is specific to cyanobacteria biomass, a potentially toxic algae. We only have about 1,000 measurements of those, which can be a little bit limiting during training. We also have 5,000 measurements of total suspended solids, shown in the bottom left, as well as colored dissolved organic matter, shown in the bottom right, proxies for nutrient and carbon availability. These again represent a wide range of different values, showing a wide range of different combinations of these different biogeochemical parameters represented in our data set. Okay, so we have this large globally distributed data set. We know we want to use a machine learning model to be able to represent and retrieve estimates in globally distributed inland and coastal waters. So we have to choose a machine learning algorithm. Well, the algorithm that our group chose was termed mixture density networks, and I'll quickly go over that algorithm here. First, as input, we take the remote sensing reflectance, or the ocean color signal, basically just the color of the water, at the wavelengths for any specific sensor. Here it's shown for either HICO or Prisma, so it's hyperspectral. This ocean color signal then goes through band ratio and multispectral algorithms that typically work well for, say, chlorophyll estimation. And these are then input into the standard weights of a neural network after going through a normalization phase where they're scaled between -1 and 1. What separates mixture density networks from typical machine learning algorithms is the output layer. This output layer is a set of Gaussians, which represent the probability distribution function for any given output product, for example, chlorophyll A. These Gaussians have a mean, standard deviation, and probability associated with them. And when we use a combination function, we can select the most likely value of chlorophyll for the input ocean color signal instead of the average value, which is what typical machine learning algorithms do. We can also simultaneously estimate all of the different products that we're interested in, including chlorophyll A, phycocyanin, TSS, and CDOM, as well as the absorbing IOPs like absorption due to phytoplankton, CDOM, and non-algal particles. And again, this was for those hyperspectral instruments. So product availability depends on the spectral availability. So I'm visually just going to try to show you why mixture density networks are well suited to solve the non-unique inverse problem. On the previous page, I showed that we have this sort of typical neural network structure. The mixture density networks differ because they output a set of Gaussians. And these Gaussians can be used to predict, let's say, the chlorophyll A concentration, when there could be a wide range of different chlorophyll A concentrations for any input watercolor spectrum. So let's say we take a watercolor spectrum like the one shown in this graphic, and we run it through our mixture density network model. What does the output of Gaussians actually look like? So we take this water quality spectrum in. We have, let's say, a measured in situ chlorophyll A concentration shown as this black dashed line. We have a prediction from the ocean color 3 band or algorithm shown as this dashed blue line. And we have a prediction from a standard machine learning algorithm shown as the dashed red line. Again, these standard machine learning algorithms typically learn to estimate the average of all possible values of chlorophyll concentration instead of the most likely value. We can then overlay the Gaussians, as shown here, that are output from the mixture density network. Each of these Gaussians, again, has a mean, standard deviation, and probability associated with it. So we can select the Gaussian with the highest probability, take its median value, and estimate that. And for this particular highly eutrophic region, that ends up having a better estimate that matches more closely with the in situ chlorophyll A concentration represented by that dashed black line than either the standard machine learning algorithm or the Ocean Color 3 algorithm. So in summary, mixture density networks allow us to better solve this non-unique inverse problem where there could be many different combinations of chlorophyll A, PC, TSS, and CDOM concentrations for the same ocean color signal. Okay, so we have this really large data set and we've picked out a machine learning algorithm that we think is going to work really well for this region. We can then do a 50-50 split from our in situ data, use half of the data for training and half of the data for validation, and then compare the results between our mixture density network and operational algorithms. And that's what's shown here. So on the y-axis, we have the estimated chlorophyll A concentration from our algorithm applied to in situ remote sensing reflectance data that's been resampled with the sensor spectral response function, in this case, MSI. And we compare that to estimates from standard operational algorithms like the Gillerson two-band algorithm or the blend algorithm. As you can see from this graphic, mixture density networks are able to achieve lower error, lower bias, higher slope, and lower root mean square log difference over a wide range of different chlorophyll values. But this is in the idealized scenario where we're estimating based on in situ data where the MDN has seen half that data. The actual accuracy when we apply it to satellite instruments is reduced. So shown here, we have MSI matchups. So they're same-day matchups where MSI, the Sentinel-2 satellite, or the imager on the Sentinel-2 satellite overpassed while the in situ chlorophyll A concentration was being measured. And on the y-axis, you can see the MDN retrieved chlorophyll A from MSI. We have very similar error metrics where MDSA is the error that we saw previously. Previously, it was about 28% when we were looking on in situ data, but once we start looking on satellite retrieved data, when we had to remove that atmospheric signal, the accuracy drops down to about 94%. So that means that the residuals from atmospheric correction substantially reduce the accuracy. However, what we can see from this plot is that the MDN matchups still well represent a really wide dynamic range from 0.1 to about 100 milligrams per meter cubed of chlorophyll A. So this means it represents anywhere from very oligotrophic waters to very eutrophic waters. So do a good job capturing the full bloom life cycle. Shown on this page, we have MDN-derived products for Lake Erie, and we can see that they're spatially consistent. So the products you see in the bottom left are actually from Olchi instead of MSI. So Olchi has more bands. We expect it to do a little bit better in terms of product accuracy as well as atmospheric correction for these regions. And we're using it here just to look at the spatial consistency of our mixture density networks when applied to atmospherically corrected data. In the top left, you can see the chlorophyll A concentration. Next to it, the total suspended sediments. In the bottom left, absorption due to CDOM. And the bottom right, phycocyanin, which again is a proxy for potentially toxic cyanobacteria. Spatially in the, let's see, if we look at the chlorophyll A map, in the top left, you can see the Detroit River plume, which has really low concentrations of chlorophyll A. In the western section of the image is Maumee Bay. You can see there's higher concentrations there. In the southernmost section, you can see really high concentrations in Sandusky Bay. And spatially, this lines up with what we've seen historically from this region. We can also look at matchups with in situ measurements that were taken, though I won't cover that here. And in general, they line up quite well. Not only can we produce products with our algorithms, we can actually also produce uncertainties, and these uncertainties demonstrate the model's confidence in its own estimates. So spatially, they're very consistent. They don't have dramatic artifacts, and they line up with what we've seen previously. But what happens when we take a temporal time series of these products? So I did that leveraging, again, Olchi, which has this fine spatial, spectral, and temporal resolution combination. And shown here, you can see a time series of a harmful algal bloom, which starts in Maumee Bay and interacts with the Detroit River bloom. So right now it's March. As it turns into April, May, and June, this bloom starts to develop. Here it's interacting with the Detroit River plume, and then we can see it sort of reset again. So temporally, we can see strong consistency in our products and actually see some hydrological interactions in these regions. So I was focusing on chlorophyll A, but we can also look at phycocyanin, which again is this marker for potentially toxic cyanobacteria. And you can see really high PC in the Sandusky Bay and Maumee Bay in the months of July and August, suggesting there might be cyanobacteria there. So now I'm going to jump to how stakeholders or users like yourselves can apply our models to preprocessed satellite imagery. The tutorials can be found in the QR code linked below. If you have any issues with it, you can also email me at the below email. But just to give you a quick overview of what's going to be included in this demonstration, first we will import some packages as well as functions from those packages, including specific functions from the MDN itself. We will then visualize pre-corrected using Acolyte Olchi imagery and learn how to plot those with some of our own functions. We'll then retrieve the RRS from this pre-processed atmospherically corrected tile. These functions can work with Acolyte or L2Gen or even Polymer. We'll then generate predictions from the RRS. So we'll generate predictions for, say, TSS or chlorophyll A. Then we can also generate uncertainties from these RRS and visually look at them and analyze the imagery. And finally, we'll do the same thing, but for L2Gen corrected PACE imagery so that we can see some of the more advanced products. All right, now we'll jump into that tutorial. So now I'll present a demonstration of our Jupyter Hub notebook called Leveraging Mixture Density Networks to Generate Biogeochemical Parameter and IOP Maps. This is one of three that we have available. The first two cover generating products from in situ measurements and generating uncertainties, and they take a little bit of a deeper dive into how these MDNs work. But today I thought I would present on how to generate product and uncertainty maps, as this is often what users are most interested in. If you have any questions on these, you can email me, Arun, or Akash directly. So the first thing we're going to do in section 1 is import the required packages and environment settings. So we can run this first block of code. This imports the Python packages needed for the notebook. They're pretty standard packages like numpy, os, pathlib, zip file, and matplotlib for plotting. And then there's some packages that we've written ourselves as part of the MDN package, which is installed via the instructions at the QR code that you saw during the presentation. I won't go through those instructions here, but if you have any issues, again, please email me. These specific MDN functions, I'll briefly go through here, and I'll dive into their exact uses a little bit more as we get into the actual code. These functions include get sensor bands, which when you provide it with the sensor name, for example, Olchi will get you the required bands for the MDN sensor. Get tile data pulls the bands as well as RRS data from a given tile path so that we can get the RRS from a preprocessed netcdf. Download example imagery we wrote specifically for this Jupyter Hub notebook. It pulls data that we have already atmospherically corrected so that users can run this notebook without having to do that atmospheric correction themselves. We have find RGB image, which pulls the red, green, and blue bands and outputs that, which we can then use as input to the display set RGB, which displays the satellite's RGB image so that we can look at the image itself, identify the aquatic regions and cloudy pixels and land pixels. We can also get tile geographic info. This pulls the extent, like lat and longitude for the given image. We can then run the RRS data that we pulled using get tile data through mapcube MDN full. And then from those predictions that it gives from the MDN, we can overlay the RGB image and the MDN products to get final maps for both uncertainties as well as the products. And this will make more sense as I jump into the examples. The next block of code that we run right here just sets the display parameters for matplotlib. I won't go into that in detail. In section 2, we just download an ULCI image that, again, I've already atmospherically corrected. If you were to do this yourself, you'll have to provide your own atmospherically corrected data and just switch out the tile paths below. So here we're setting the sensor to ULCI, and the date location just allow us to pull the correct example imagery. I've already downloaded that tile locally, so and it's displayed at this tile path right here. After running that, we can then display the RGB image using display set RGB, and that image looks like this, which I'll go through in a second. It just takes as input the tile path and sensor. You can change the size of the figure as well as the title, which we pull from the sensor and location and date that we specified previously. And it outputs the image as an RGB image, which we can use for future algorithms. So this can serve as the base map for future functions that we've written. So if we just like a look at the RGB image itself, you can see that this is an image from ULCI of San Francisco Bay on March 16th, 2019. Let's see, this is the Pacific Ocean. You can see it's pretty blue, meaning it's probably dominated mostly by chlorophyll. It probably has low concentrations of TSS and sediment. As it moves into the San Francisco Bay, you can see it turns maybe greener. Maybe there's higher concentrations of chlorophyll there and higher phytoplankton biomass. As you move up to the northern section of San Francisco Bay, it turns sort of a yellowy color, suggesting maybe there's higher concentrations of suspended sediment there from the Sacramento River and San Joaquin rivers. And if we look at the southern section of the bay, you can also see these little areas, which are either bright green or bright orange or bright yellow, and these are the salt flats, which are separated from the bay. So there's a wide range of different optically distinct regions represented here in this image. You can also see some clouds in the bottom left. Okay, so we have the tile data, we have the RGB image. Now we can pull out the remote sensing reflectance data with this block of code. Using get tile data with the tile path specified and the sensor specified to ULCI, it'll pull out the bands and RRS required by our MDN prediction function. We can also use get tile geographic info from the tile path to get the lat long and extent for the image. And then we can plot the RRS data for this specific band, just for the first band, just to check that we plot the RRS correctly. Indeed, we can see the RRS is pulled out for all the aquatic regions, and there aren't any RRS pixels for land or clouds. Now that we have the RRS data, we can move on to generating predictions from the MDN using our MDN cube or map cube MDN full function. So I'll run that while I just go through this function. You can see it running in the bottom. You can mute this output, though here we've included it just so you can see that it is running. So from the MDN package, we can pull map cube MDN full as well as get args. These allow us to map the model predictions and uncertainties and define the arguments that select the correct model. So for example, in kwargs here, we set the sensor to ULCI, the products to chlorophyll, TSS, and CDOM, and we set the model UID, which is the name of the model weights that we actually use. You don't have to change that for now. If you have questions about setting the model UID for specific sensors, you can again message me. After we run get arguments, which pulls the correct arguments for this set of keyword arguments, we can then run mapcube MDN full on the arguments and RRS that we've already pulled, as well as the bands. There's a few different parameters that we can set, like landmask, which we set to false here as we just use Acolyte's landmask. There's also figure subsample, which allows us to subsample the image so we could reduce the spatial resolution, basically. This might be useful if you want to plot an MSI image quickly and it has too high spatial resolution and it takes a little too long on your computer to run. We also have scalar mode set to invert. This doesn't need to be changed, but it just basically puts the model uncertainty values into the same scale as the model predictions. Block size is set to 10,000. This is one that you might want to change, as this controls the size of the, or the number of the RRS that are processed at a single time. So reducing it allows you to run this on a personal laptop, and increasing it allows you to run things faster if you have more RAM available. And the uncertainty mode is set to composite, so instead of providing uncertainty bounds like low and high uncertainty estimates, it just produces a single model uncertainty. All right, it looks like this ran in the time I took to explain it, and you can see the outputs for each block of 10,000 RRS. Again, you can mute this if necessary, but it looks like it finished running. We can then, from the model predictions, plot graphics for chlorophyll, as well as for TSS and for CDOM. And then we can start to visually sort of analyze these products. So these plots are created using overlay RGB MDM products, where we take the RGB image as input. The model predictions, which were output by the previous algorithm that ran, our mixture density network model. We pull all of the 2D information, and we can pull the slice that corresponds to chlorophyll. And how we change it is we just change from chlorophyll to TSS or to CDOM, depending on the product that we want to plot. For this chlorophyll product, we have this string here that gets set that we can use as the title, and we can put prediction ticks from 10 to the 0 to 10 to the 2. So these are in log scale. And we can also set the figure size. And that produces these plots with the RGB image background and the MDM products overlaid. Okay, so we saw darker waters in the Pacific, and that corresponds to lower concentrations of chlorophyll. That checks out. We saw these really bright salt flats, which have high concentrations of chlorophyll. That also sort of makes sense. And we see a little bit of slightly higher concentrations of chlorophyll in the northern section of San Francisco Bay and the Sacramento and San Joaquin rivers. This generally tracks with what we saw visually in the RGB imagery. We can also look at the total suspended solids map that we produced from that same image. If we follow this from the Pacific Ocean, you can again see lower concentrations in the Pacific, which makes sense with how we understand the region. Higher concentrations seemingly, coming out of the San Francisco Bay. And as you move into the northern section, you see really high concentrations, which came from these two rivers, which again tracks with our understanding of the region. And we can do the same thing for CDOM, where again you see lower concentrations in the Pacific Ocean and higher concentrations in the northern section of this bay. Not only can we get the products, but we can also visualize uncertainties, again using this overlay RGB MDM products function. But now we just also set the image uncertainties using the output model uncertainties for the corresponding slice, for example, chlorophyll in this example. So we can run that for chlorophyll, TSS, as well as CDOM, and we can start to see the model's confidence in its own predictions in the same space as the chlorophyll A predictions itself. So you can see the regions where there's higher uncertainties. Often you'll see some of these higher uncertainties near cloud, or maybe some of the atmospheric correction failed, and in the really high concentration chlorophyll estimates in these salt ponds. which generally tracks with what we would expect for this region. Finally, we can move on to section 6, where we're generating predictions from hyperspectral PACE/OCI imagery. We can, again, just download some example imagery for a day where I've already corrected this imagery from an image of Lake Erie. Then we can display this RGB image from the PACE image that we've, again, already atmospherically corrected. You can see it has darker waters in the eastern section, brighter waters in the western section in Sandusky Bay, which again tracks with our understanding of this region. And again, lower brightness from the Detroit River plume. We can then do all of the same functions, or use all the same functions that we used previously, like get tile data, get the tile geographic info to get the RRS and lat long, plot the RRS, and then use MapCube MDN full on that RRS to get model predictions, uncertainties, as well as the slices. So I'll run that in the background here. And again, you can see it running with this process bar. That'll run in the background, but we can already look at some of the visual imagery that I've produced previously. In the top here, you can see MDN estimated for chlorophyll A, PC, TSS, and CDOM. So this hyperspectral algorithm allows us to pull out additional products like phycocyanin, as well as more advanced products like IOPs, which you can see in the bottom. But spatially, I can just quickly go through what we expected to see, where you can see lower concentrations in the Detroit River plume and the eastern section of Erie, and higher concentrations in Maumee and Sandusky Bays. For PC, this is in May, so you would expect there to be lower concentrations of phycocyanin as these harmful algal blooms have not started yet. And you can again see that in most of this image, though there's a little bit of higher concentrations in Maumee Bay. You can see similar spatial distributions for TSS as well as CDOM. We can also look at the more advanced products like absorption due to phytoplankton, non-algal particles, as well as CDOM. Shown here is MDN estimated APH at 450 nanometers for PACE. You can also see different wavelengths like 490, 550, 620. This one often corresponds to cyanobacteria, and 670. And we can also look spectrally at absorption due to CDOM, where we've just plotted 440 nanometers, 490, 560, 650, and 675. And we did the same thing for non-algal particles. So you can see that with this hyperspectral data, we're able to represent a wide range of additional products. Thanks for listening to my demonstration. I'll be happy to field any questions you might have during the question and answer period after Mita provides some summary information. Thanks.
Thank you very much, Ryan, for your excellent presentation and demonstration of the code, and also for sharing the code with everyone. That brings us to the close of today's session. To summarize, we started with an overview of pre-processing top of atmosphere radiances from satellite images. We would start with level 1 satellite data. We saw that there are several data portals where different satellite and sensor level 1 data are available. Then get atmospherically corrected level 2 remote sensing reflectances from the TOA data. And these remote sensing reflectances can be used as inputs to the MDN models. Then Dr. O'Shea provided an overview and demonstration of using MDN models to derive water quality parameters. Specifically, the models are trained using a large globally distributed dataset of in situ water quality measurements and co-located hyperspectral measurements from a wide range of optically complex regions, and sharing many of these samples with Gloria data. The MDN approach is well suited for addressing non-unique inverse problems like ocean color remote sensing due to its output layer. The models retrieve key water quality parameters depending on spectral availability, including chlorophyll A, total suspended solids, colored dissolved organic matter, and phycocyanin, along with their associated uncertainties. Similar models were developed to support multiple multi- and hyperspectral satellite imagers, including MODIS, VIIRS, Landsat 8 and 9 OLI, Sentinel-2 MSI, Sentinel-3 OLGI, and PACE OCI. This brings us to the conclusion of this webinar series. To summarize, we started with an overview and demonstration of the satellite-based tool for rapid evaluation of aquatic environments, or STREAM, web tool and API in part 1. We saw how to map water quality parameters including chlorophyll A concentration, total suspended solids, and Secchi disk depth. The data are available from 2024 to near real time in coastal estuaries and inland lakes across the US at 20 to 30 meter spatial resolution, and these data are derived from Landsat 8 and 9 and Sentinel 2A, 2B, and 2C imagery. We saw how to search and download water quality products using the STREAM web tool and API, and then we examined time series of water quality parameters in the region of interest using QGIS. Today in part 2, we had an overview and demonstration of the Aquaverse algorithm. So introduction to the MDN models to derive water quality parameters, and access and application of MDN based model code to retrieve water quality parameters. Finally, STREAM web tool and API and the MDN code are all open source, and you can download and use the code as we saw today from Dr. O'Shea. There is one homework assignment, and it's posted on the training web page today. Answers must be submitted via Google Forms, and the homework is due on 10th March. Certificate of completion will be awarded to those who attended all live webinar sessions, would complete the homework assignment by the deadline, and then you will receive a certificate via email approximately 2 months after completion of the course. Once again, we thank our guest speakers, William Wainwright and Ryan O'Shea, for their presentations and demonstrations describing how water quality parameters are derived from remote sensing and then making these data available through STREAM website. Trainer's contact information is provided here, and you can always contact RSET. RSET website and RSET YouTube link are given here. All the training materials, this training and previous trainings material can be found here. For questions, comments, or to share how you have applied our trainings to your work or studies, email nasa.rset@gmail.com. Join our quarterly newsletter to stay up to date on our latest trainings, and the steps are given here. Just send an email with no subject line to this email address and follow the instructions sent in response. Here are some useful resources. And with that, we thank you all for attending today's session, and we will go to the question and answer session now. We'll start with the question and answer session. And once again, we thank our guest speakers, William Wainwright and Dr. Ryan O'Shea. We'll start with question 1. How much does spatial resolution affect the processing time of the MDN? And you can unmute Ryan and answer the question.
All right. Thanks. I'd say most of the variability comes from the number of aquatic pixels in the given image. So for the same area, MSI, if you're using it at the 10 meter spatial resolution, would have almost 900 times more pixels than, say, OLGI at 300 meter resolution, and therefore take that much longer to process. But really, it's a function of the number of available aquatic pixels.
Great, thank you. Next question is, what is the difference between PC and TSS?
Yeah, sure. PC is defined on slide 20 in the presentation. It stands for phycocyanin. Phycocyanin is a pigment that is specific to cyanobacteria biomass, which is a potentially toxic algae. And TSS stands for total suspended solids. These can be things like sand, which has been suspended in the water column.
Next question, is there any reason to select 50-50 training and validation dataset? We usually consider 80-20 or 70-30.
Sure, we have a long response to this question in the Q&A document. But the short answer is to ensure statistically robust and representative validation across the range of observed conditions. So we adopted this 50-50 split. We have a pretty large data set, so we're able to split it and get robust validation metrics. Additionally, this is really our idealized error metric, I guess, that we're reporting here. We have multiple other ways of testing these algorithms. One of them is a regional split, where we'll split the data regionally. So we'll train on everything except for one region and test it in that region, and then report the error metrics there to get an out of training data set estimate. That usually has like 50% uncertainty or residuals. And then the second way, which I showed here today, was we'll also try to get matchups with the satellite sensor for the probably most realistic error metric of what you'll actually see from satellite imagery.
Thank you, Ryan. Question 4 is, just to be sure, Stream uses TOA radiances for satellites as input to the MDN model to derive water quality parameters?
Sure. So Stream actually has two different MDNs. One MDN retrieves the remote sensing reflectance or ocean color from the TOA radiance. And then we actually have a second MDN that retrieves water quality parameters from the remote sensing reflectance or ocean color signal. So most of what we've published so far has been on that second MDN, where we try to retrieve these water quality parameters, things like chlorophyll A, TSS, Secudis depth, from the RRS. But we are rapidly trying to get out a publication to retrieve the RRS from the TOA radiances as well. And we expect in the next, 6 months to have that MDN available online, but we'll keep you posted.
Great, thank you for that reply. And if you go back and see some trainings that ARSA did, in those trainings, CDAS or CSSW have been used. That's also NASA software, and soon Aquaverse will be available as well. Question 5, the MSI scatter plots show a classic heteroscedasticity pattern of residual, there are statistical corrections that can account for this and provide better predictions. Has anyone tried that? The residuals are clearly a function of the X variable.
So this isn't exactly my area of expertise, but somebody else in my group wrote a response, which I will read here. The MSI scatter plot should not be interpreted in the classical regression sense. In this case, the x-axis does not represent an independent predictor variable upon which the y-axis depends. Model predictions are generated from the input RS, whereas the x-axis represents independent in-situ measurements used solely for validation. We acknowledge the presence of systematic bias and range-dependent error behavior in certain portions of the dynamic range of the water quality variables. These trends are consistent with the heteroscedastic characteristics of the dataset and are an active area of ongoing model refinement.
Thank you. Question 6 is, as MODIS temporal resolution is higher than Landsat Sentinel, is there a way to use it as a support tool that works whenever there are clouds or glint effects and getting a higher amount of images?
Yeah, so this is again another active area of research. How do we harmonize between these different satellite sensors and combine together their outputs. So one thing we tried to do with these mixture density networks was apply them to the available satellites that we had access to. And I showed in one of the earlier slides all of the different satellites that we do have MDNs that that support these different satellite sensors. These MDNs are trained on similar data sets, but each of these sensors does have different spectral characteristics. So what we end up seeing is that these MDNs generally produce pretty harmonized retrievals for the different products, including chlorophyll between the different sensors. However, long term, we're trying to attempt to further improve harmonization between these sensors, specifically Landsat, Sentinel-2, and Sentinel-3, by leveraging even more advanced machine learning models. So I guess the short answer to that question is yes, you certainly can use our models to fill in and build time series, and we're trying to improve that in the future.
Thank you. Question 7. Can planet data be used for MDN or is among the coming soon?
Yeah, actually, we do have an MDN available for chlorophyll A and Psyche from Planet SuperDove. I provide a link in the Q&A text. However, it's not currently supported as part of the tutorials or GitHub. We do plan to add it soon, probably in the next month or two, as we are rapidly adding in these additional sensors that we have models for but aren't quite supported by the tutorials just yet. So feel free to email me, and when we push the updates to our GitHub, I can send an email back.
Wonderful. Thank you, Ryan. Our next question is, have the MDN results been compared with the derived products from Ecolite?
I'm not sure exactly what algorithm Acolyte uses, but I guess the short answer to this question is no, we haven't done that direct comparison. We have done comparisons to other, operationally available algorithms where we download the products from websites like the Ocean Color V4 algorithm, as well as Cyan, for regions where we have a lot of in situ data like Lake Erie and the Chesapeake Bay. And in general, we've seen the MDNs outperform these algorithms across the wide dynamic range that's represented, particularly in Lake Erie. So for bloom formation in particular, they're quite robust.
Next question is, can this code be used anywhere? Do we have to change the input, which depends on the zone that we study?
Short answer is yes, it can be used anywhere. The model only relies on RRS, or the remote sensing reflectance, the ocean color signal, as input, so it can be used in any region. There aren't any other inputs.
Question 10. Currently, I'm working on estimating TSS using surface reflectance data. Since high TSS in situ data are not available for training, my model predicts low TSS values during extreme events, even though I know the actual TSS concentrations should be much higher than the estimates. Could you please suggest how to address this issue?
Let me think about that one for a second. In this case, are the TSS being estimated from the MDN? And I guess how high is high for the TSS values? The short answer, I guess, is that if you did have high TSS values with RRS available, you could potentially either retrain the MDN or apply a transfer learning approach where you take the MDN that we have and then just retrain some of the later layers using this in situ data. Yeah, I think that would probably be the best approach.
So Ryan, would you say that because this was trained, MDN was trained with like a large range of values, it may perform better than other retrieval schemes for TSS?
I would expect it to perform pretty well for the region, or for the concentration ranges that we have available, but not necessarily outside of that. We do have limited data at the higher ends.
Right.
So that might be a limitation for accuracy.
Thank you. Question 11, is it possible to fine-tune MDN using in situ data?
Yeah, that sort of builds off that last question. The short answer is yes, it is possible, particularly using transfer learning. I think we've tried that in our lab before. I do think we have some code for it. We don't currently support that via our tutorials, but if you send me an email, we can set up a video conference or something, and we can discuss how to do this yourself.
Great, thank you. Ryan, question 12. The uncertainty maps seem to look very similar to the water quality maps. High WQ estimates equals high uncertainty. Does that mean that the model can be less reliable at higher values?
I guess I'd say that the bulk value of uncertainty is higher for higher values, and we can use relative uncertainty or percentages to identify outliers in terms of prediction performance.
Mhm. Next question is, can uranium contamination in surface water be estimated?
I don't have any experience with mapping uranium contamination, and yeah, I don't think we've used that previously or seen data from that.
Question 4 is, do you have any guidance or recommended resources for pre-processing the RRS inputs? Specifically, I'm interested in MSI and OLE.
Yes. So the I guess atmospheric corrections that from our experience we've seen work well in eutrophic waters is acolyte, and more oligotrophic waters is L2 Gen or CDAS. Also coming soon, we are producing our own in-house aquaverse atmospheric correction method. We are working on a paper for that and should have that out in the next sort of 6 months. So stay tuned.
Next question is the sample data set histogram shown earlier, what is the temporal range and the spatial extent of the data?
Sure. It's similar to that of the Gloria database. We've taken the Gloria database and sort of extended it with our own in situ data as well. I'm not sure exactly on the temporal range, but much of the data is from the last 20 years. And spatially, most of the data from that map I showed, I don't know the slide number, but I do have that graphic where it shows about 80% of the data is from North America and Europe, and maybe 20% is from the rest of the globe. We are trying to build that out. If if you have data set from underrepresented regions, we'd be happy to incorporate that into future versions of the MDN model. And theoretically, that would improve the MDN's retrievals in those regions. So again, yeah, feel free to send me an email message if you're interested in that.
Thank you. Next question is there no atmospheric correction embedded in the MDN? Do you suggest just starting with L2 data?
Yeah, that's correct. So the MDNs that are available on the GitHub tutorial right now just start from the level 2 data. So I'd recommend something like L2 Gen or Acolyte. Again, we are working on developing in-house atmospheric correction models. We have some that work really well, but we have to get those publications out, and then we can add those to the GitHub tutorials. We're planning to do this in advance of Ocean Optics, which is in, I believe, September, where we'll host a tutorial there.
Next question, is it possible to apply these models to earlier versions of Landsat, such as Landsat 5 or 7? What would be the limitations?
Let me think about that for a second. I think and somebody from my group can correct me if I'm wrong, I believe we have applied some of these models for Secchi disk depth retrieval from Landsat 5 and 7. But I don't think we've done that for chlorophyll A or TSS. So I'm not sure if we've explored that fully. I'm not sure how the SNR for these sensors would support retrieval of chlorophyll A products, but that's definitely something we'd be interested in exploring.
Thank you. Next question is, are there any matchup studies for MDN-based predicted TSS and CHLA?
Not published. However, we are doing that in-house. We are funded as part of the PACE Science and Applications team. We are working on one such publication where we have matchups for chlorophyll A and phycocyanin across oligotrophic waters in the U.S. that, I don't know, hopefully will be out in the next year. And then also we have an in-house validation data set from the PACE validation team, and those seem to do really well when compared to OCV4 in more, again, eutrophic inland and coastal waters. So short answer is yes, we've done these studies, but they're not published yet.
Thank you. Next question. What is the accuracy of the MDN network?
That's a difficult question to answer because there's a few different accuracies that you can report and for multiple different satellite sensors. So it really depends on the satellite sensor's spectral capabilities, as well as its SNR, the accuracy of the atmospheric correction, and if you're applying this algorithm to in situ RRS without any atmospheric correction, or if you're applying it to satellite imagery. If you are interested in the accuracy for specific sensors, we have a wide range of publications that are cited in the tutorials at the bottom. You can look through those, and in general, we try to publish the accuracies in each of those different scenarios for the different sensors that we support.
Thank you. Next question is there a reason why you did not compare with other available machine learning algorithms?
Yeah, I guess typically we try to compare with the sort of more operationally available algorithms. And we've generally focused on those. We'd be happy to compare against other machine learning algorithms if there's any specific one that you're interested in with any data sets that we might have.
Thank you, Brian. Question 21. Is the same network used for all sensors?
So for the multispectral instruments, we typically try to use the same MDN network architecture with the same number of layers and neurons. However, when we move to hyperspectral sensors like EMIT, Prisma, and PACE, we had to increase the number of neurons for the larger amount of inputs as well as outputs, because now we were inputting hyperspectral data and we're outputting more complex products like absorption due to phytoplankton, which is spectrally variable. So we have something like 50 inputs and 70 outputs. So the short answer is for the multispectral instruments, yes, it's very similar. And for hyperspectral, we had to increase the number of neurons in the network. But we use MDNs for both. Sorry.
Yeah. So next question is, I didn't understand that one MDN retrieves RRS for IOP, ocean color properties, and the second MDN retrieves water quality parameters. Isn't it supposed to be the same?
Yeah, so for each sensor, they have different spectra that are supported. So for the multispectral instruments, typically what we retrieve is chlorophyll A, TSS, and CDOM, also Secchi depth sometimes for the models that we support. But for the hyperspectral instruments, we started adding in the IOPs as well. So not only do we retrieve chlorophyll A, TSS, and CDOM, but we also simultaneously retrieve absorption due to phytoplankton, colored dissolved organic matter, and non-algal particles. Yeah, so it depends on the sensor which products we currently support, if they do retrieve them simultaneously.
Next question is, which bands are the network using for chlorophyll A and the other parameters?
Yeah, again, it depends very much on the sensor that we're talking about. Typically, we try to keep the bands in the, I believe it's 415 nanometer to about 750 nanometer range. That varies a little bit sensor to sensor. The exact wavelengths can be found in the tutorials using the get censored bands function for the specific sensor. But we try to use all the bands in that range typically.
Next question is there a document that lists the water bodies where the MDN is trained and validated?
There's an individual document. However, we do have, again, a wide set of publications available. These are cited at the bottom of our tutorials. And in each one of those publications, we go through very similar training where we do the 50-50 split. Sometimes we do a regional split as well. And then finally, we try to do a matchup analysis for each one of the sensors. And in that, we do also talk about the sort of spatial distribution of that data.
The next question is there, can it distinguish between oil spills and HABs over oceans?
We don't have experience using these MDNs for distinguishing between oil spills and HABs.
Okay, the next question is how do NASA ocean color and inland water quality algorithms manage seasonal spectral shifts caused by monsoon-driven turbidity, CDOM variations, and changing solar geometry without introducing systematic bias in long-term time series?
Let me think about that one for a second. Yeah, I think this is a really good question. I think I'd have to think on it a little bit before providing an answer. I'll try to do that over the next week and get back to it.
Great, thank you so much. The next question is why was the in situ chlorophyll A data from USGS, which covers the United States, not used?
Let's see. Does that data set include RRS as well as chlorophyll A? If so, we'd be happy to add it in. I'm not entirely sure if it does. Yeah, if it does include RRS, we'd be happy to add it in when we retrain these models. If it is just chlorophyll A, we probably have pulled some of that data for validation. But we can't use it for training with the MDN since they require the RS as input.
Question 28. Does Stream recommend regional recalibration of retrieval algorithms, or does it rely on globally parameterized models with deviation correction layers?
We do not currently recommend regional recalibration of these algorithms. Usually what we would like to do is just keep adding in more and more data globally. So again, if you have in situ data with RRS and the parameter of interest, and you're willing to share it, please send me an email and we'd be happy on future model updates to include that data, which theoretically should increase the accuracy of the model for that given region. The model is globally trained. We don't deviate at all based on the region. So the same model gets applied just to the RRS no matter what region we're in.
Thank you. Next question. Can the MDN models be used for volatile and non-volatile compounds as part of water quality predictions? In the case of nitrates, phosphates, ammoniacal nitrates, and other ions, could MDN be used? If yes, in general practice, how many in situ observations are we looking here to train MDN models?
Oh, I see. The MDN can only be used for, well, I guess the MDN from these satellites with multispectral or hyperspectral data in the visible and near infrared range can only be used for products that change the ocean color signal. If you have RRS with different concentrate, I don't know specifically how these different compounds change the ocean color signal or if they do interact. If you do have a data set of RRS with different concentrations of these different products and they do change it, then we could try to train an MDN to do that. For how many in situ observations? Again, that sort of depends, but I guess we've used in practice as little as 1,000 samples to train our PC models. So that represented a wide range of different phycocyanin concentrations pretty well. But I don't know if we've looked at an exact minimum.
Next question, can MDN model run on wetland water quality monitoring?
Let me think about that for 1 second. If the wetlands are optically deep, meaning that we can't see the bottom reflectance, then theoretically, I believe they should work just as well for retrieving chlorophyll A or TSS. However, if you can see the bottom reflectance, that is going to change the ocean color signal or the signal retrieved by these atmospheric correction algorithms. And we really haven't tested the model in these regions.
Thank you. Next question is, should input L2 data be graded before using the MDN, or does it not matter?
1 second. No, I think the answer is no. It doesn't matter. It doesn't have to be gridded. Yeah, the MDN just takes as input the remote sensing reflectance in basically an array. So as long as you can get it into the format similar to that, that's applied in the tutorials, you should be good.
Thank you so much. It looks like there's a lot of interest and we had a lot of great questions. We will be posting our Q&A documents on the training website in a week or so for both the sessions. Okay, I think there is one more question. How was the RRS obtained for Gloria and other datasets used? Were they downscaled from any satellite data to the location of the sample site? If so, can I do similarly for my in situ datasets which only have chlorophyll A?
Sure. So the Gloria data set, and we actually use an augmented version of that, but it has very similar properties. Has hyperspectral RRS and a few different in situ products, including chlorophyll A. We take that hyperspectral RRS and resample it with the sensor's relative spectral response function, and then we train the models using that resampled RRS. You could do that with your own in situ data set if you have chlorophyll A with RRS. If you only have chlorophyll A, you wouldn't be able to retrain.
I'm currently working on a coastal water quality study area. This area covers a small region and I want to use Stream, but Stream does not support this area with data. What can I do?
You can email us. We are trying to support users that are interested in using our products. We do have some processing limitations that we're trying to work through, but if you email us, we can try to add a specific tile in that you might be interested in.
That's fantastic. Thank you so much. Are these data accessible in Google Earth Engine?
No, we have not included these data in Google Earth Engine yet. We do not currently have plans to.
Hello, I'm from Mexico and I would like to use this tool in my country, but I saw it's only available in the US. Can be the possibility that we can use in Latin America?
Sure. Again, yeah, feel free to email me if there's a specific site that you're interested in, and we can try to add that to stream. Again, we do have some processing limitations, but if it's only one site, we should be able to do that. If you're talking about stream, if you're talking about the MDNs themselves, we do have data from, again, around the globe, and we've used that in training. As long as you have RS data from a satellite using the tutorials that I showed here today, you should be able to produce your own product maps for that region. We're also, as a part of the Landsat science team, we're also adding in data from some Latin American countries, including Mexico, and trying to validate our algorithms in these regions.
This is one of the last questions we can take. In highly turbid Indonesian tropical river systems, such as the Kapuas and Mahakam in Kalimantan, where CDOM and suspended sediments are both elevated, how does the MDN handle spectral ambiguity between chlorophyll A and CDOM absorption in blue bands?
Sure. So we typically treat the MDN more as a black box. We don't really go into like the layers to see how it is estimating chlorophyll A and CDOM. But the way it works is it's an empirical algorithm. So we have again a large data set with a wide range of chlorophyll A, CDOM, TSS values represented. So if we have similar chlorophyll A and CDOM combinations, we would expect it to perform relatively well. If we don't, it might not. But then you could, again, if you have in situ data you're willing to share, you could email me and we could add it into future trainings in the model.
Great, thank you. I think we are almost at the end of today's session. Thank you all for attending this training. And once again, we thank our guest speakers for their valuable time and information that they've provided. Also thank the RSET team for their coordination and help in putting this training together. And we hope to see you at our next training and look forward to get survey results from you and feedback from you. Thank you.